Welcome to the SWAYAM NPTEL course on food microbiology for safe and sustainable food systems. We were discussing beneficial microorganisms in food, and today in this lecture we will be discussing biochemistry of some beneficial traits, and we will be covering in this lecture the introduction, mechanism of transport of nutrients, transport and metabolism of carbohydrates, transport and metabolism of proteinaceous compounds and amino acids, and lastly the transport and metabolism of lipid compounds. The introduction. The beneficial microorganisms metabolized some of the components present in the starting material, for example, foods such as milk or meat, to produce energy and cellular materials and to multiply. In the process, they produce some end products that are no longer necessary for the cells, and therefore, these are byproducts and are excreted in the environment. Some of these byproducts impart unique characteristics, mostly texture and flavor, to the remaining components of the starting material. Some of the byproducts of fermentation can also be purified and used as food additives. Before the substrates are metabolized inside microbial cells, they have to be transported from the outside environment. It will be beneficial to recognize in brief the cellular components involved in the transport of these substances. The important substrates available in the starting materials of fermentation include several carbohydrates, proteinaceous and non-protein nitrogenous, that is NPA components, and lipids. The important fermentable carbohydrates in foods are starch, glycogen in meat, lactose in dairy products, sucrose, maltose from breakdown of starch, glucose, fructose, and pentose from plant sources. The proteinaceous and non-proteinaceous components. The proteinaceous and NPN components include mainly large proteins, both structural and functional, and peptides of different sizes and amino acids. Lipids can include triglycerides, phospholipids, fatty acids, and sterols. Microorganisms differ greatly in their ability to transport these components from outside and metabolize them inside the cells. Now, the mechanism of transport of nutrients. Nutrient molecules have to pass through the cell barriers, the cell wall and the cell membrane. However, in most gram-positive lactic acid bacteria, the main barrier is the cytoplasmic membrane. The cytoplasmic membrane is made up of two layers of lipids in which protein molecules are embedded, some of which span the lipid bilayer from the cytoplasmic side to the cell wall side. Many of them are transport proteins involved in carrying nutrient molecules from the outside into the cell, also removing from many byproducts from the cell into the environment. In general, small molecules such as mono and disaccharides, amino acids, and small peptides up to 8 to 10 amino acids are transported almost unchanged inside the cell by specific transport systems, either singly or in groups. Fatty acids, either free or hydrolyzed from glycerides, can dissolve and diffuse through the lipid bilayers. In contrast, large carbohydrates, for example polysaccharides as starch, large peptides and proteins such as casein, albumin cannot be transported directly inside the cell. If a cell is capable of producing specific extracellular hydrolyzing enzymes that are either present on the surface of the cell wall or released into the environment, then large nutrient molecules can be broken down to small molecules and then transported by the appropriate transport systems. Mono and disaccharides, amino acids and small peptides are transported through the membrane by different active transport systems such as primary transport systems, for example ATP binding cassettes or ABC transporters. Secondary transport systems, for example uniport, symport and antiport systems that use proton motive force, and phosphophenol pyruvate phosphotransferase, that is PEP-PTS systems. A system can be specific for a type of molecule or for a group of similar molecules, that is the group transfer, can transport against the concentration gradient of a substrate, and the transport process requires energy. In the PEP-PTS system, for PTS sugars, the energy is derived from PEP. In the permease system, for permease sugars, amino acids and probably small peptides, energy is derived from proton motive force. Now, the transport and metabolism of carbohydrates. In lactic acid bacteria and other bacteria used in food fermentation, disaccharide and monosaccharides, both hexoses and pentoses, can be transported by PEP-PTS as well as by permease systems. The same carbohydrate can be transported by the PEP-PTS system in one species and by the permease system in another species. Similarly, in a species, some carbohydrates are transported by the PEP-PTS system, whereas others are transported by the permease systems. Now, the transport and metabolism of proteinaceous compounds and amino acids. Many lactic acid bacteria used in food fermentation have active transport systems for transporting amino acids and small peptides approximately 8 to 10 amino acids long. Large peptides and metabolizable proteins in the environment are hydrolyzed by the proteinases and peptidase enzymes located mainly on the cell wall of these species. To small peptides and amino acids and then transported many by active group transport systems inside the cells. Inside the cells, peptides are converted to amino acids, and the amino acids are then metabolized differently to produce many different end products which are excreted in the environment. Some of the metabolic processes involve decarboxylation, that is generates amines, some of which are biologically active, such as histamine from histidine, deamination, oxidative reduction and anaerobic reduction. Amino acid metabolism by lactic acid bacteria and different associative bacteria during fermentation can produce diverse products, many of which have specific flavor characteristics. Some of these include ammonia, hydrogen sulfide, amines, mercaptans and disulfides. Many of them in low concentrations contribute to the desirable flavor of different fermented foods. Proper hydrolysis of food proteins by the proteolytic enzymes of starter microorganisms is important for the desirable texture of many fermented foods, such as in cheeses. A rapid hydrolysis of proteins can result in production and accumulation of some specific hydrophobic peptides that impart a bitter taste to the product, such as in some sharp cheddar cheeses. Now, transport and metabolism of lipid compounds. Many starter culture bacteria metabolize lipids poorly. However, molds have better lipid metabolism systems. Triglycerides and phospholipids are hydrolyzed outside of the cell by lipases produced by the microorganisms, releasing fatty acids and glycerol and glycerides, that is mono or diglycerides. Fatty acids can diffuse through the membrane into the cells and be metabolized. Some fatty acids are incorporated in the membrane. Hydrolysis of glycerides, especially those with small fatty acids such as butyric acid, can cause hydrolytic rancidity of the products. Oxidation of unsaturated fatty acids by microorganisms, especially molds, can produce many flavor compounds, either desirable or undesirable. That is all for today. Thank you very much.